Hello to whoever is watching this video on YouTube. As of May 12, 2020, this is my microbiology unknown bacteria presentation, number 008. For this presentation, I'll be going over the different types of test results for my unknown and how I came to conclude what my unknown bacteria is. And towards the end, I'll also go over the key points, disease, host transmission, and treatment for my unknown bacteria. So I was given a project to identify a unknown bacteria. On the left side here, as you can see, I was given a gram-negative bacteria. On the right side here, on the top is just blank media and before incubation down here. So on this page here, the picture on the left side here, this is after incubation with a top is black background and the bottom is white background. The picture on the right side here, we were told to use a lab manual as a guide just to help us compare the different test results of all the bacteria. But my bacteria that I did find was EA, the Enterobacterial aerogenes. This next slide here is just a bigger picture of after incubation, just to give you a better view of the different color results. So the first test I looked at was the nutrient gelatin liquefaction test. This test is basically tests the ability of the organism to produce an exoenzyme called gelatinase that hydrolyzes gelatin. If it tests positive, it will be liquefied. If it tests negative, it will be solid. My unknown actually tested negative. The next test I looked at was a lactose fermentation test. This test is to see if the microbe can ferment the carbohydrate lactose as a carbon source. My unknown actually tested positive. It did have acid gas production in it. The enzyme is B-galactosidase and the substrate is lactose, and the positive results did show a yellow with orange color with gas. The next test I looked at was the deoxy fermentation test. This test is used to test the organism's ability to ferment the sugar glucose as well as its ability to convert the end product of glycolysis and the pyruvic acid into gaseous byproducts. My unknown actually tested positive for this, and you can see it had acid and gas production. The enzyme is none because it's capable of entering glucose by itself. The substrate is dextrose, and the positive result shows a yellow color with gas. The next test I looked at was the sucrose fermentation test. This test is to see if the microbe can ferment the carbohydrate sucrose as a carbon source. My unknown actually tested positive for it. It did have acid gas productions on it. The enzyme is sucrose, the substrate is sucrose, and the positive result shows yellow color with gas. The next test I looked at was the endo test. This test helps screens for the ability of organisms to degrade the amino acid tryptophan to produce endo. My unknown actually tested negative for this. The next test I looked at was the H2S gas production test. This test helps determine whether the microbe can reduce sulfur-containing compound to sulfides during the process of metabolism. My unknown actually tested negative for this. The next test I looked at was the methane red test. This test is used to detect the ability of an organism to produce and maintain acid end products from glucose fermentation. My unknown tested negative for this. The next test I looked at was the Voges-Proskauer test. This test is used to determine if an organism produces acid acetylene methane carbonyl from glucose fermentation. My unknown tested positive for this. The substrate is glucose. The product is butanediol and acetylene, which is the precursor to this butanediol. The positive result shows a red color. The next test I looked at was the Siemens citrate slate test. This test is to see if the microbe can use the compound citrate as sole source of carbon and energy for growth. My unknown tested positive for this. The substrate is citrate and the product is carbon dioxide and the enzyme is citrase. The positive result shows a blue color. What that is the carbon dioxide is mixed with the sodium outside the cell, which forms the sodium bicarbonate that drives the pH, making it turn into a blue color. The next test I looked at was the nitrate reduction test. This test determines the production of an enzyme called nitrate reductase, which reduces in the reduction of nitrate. My unknown tested positive for this. The substrate is nitrate, the product is nitrate or other nitrogenous compound. The positive result shows a red color of after sulfuric acid and the, I apologize, I can't say that word, but alpha antropholamine. And the thing is that if it's a light color or it gives a little colorless, and if you look at the bottom, if you see some dust on dirt, zinc has been added into it. The next test I looked at was the urease activity test. This test is used to differentiate the organisms based on their ability to hydrolyze in urine with enzyme urease. My unknown tested negative for this. Some key points for my unknown, which is EA. EA is the genus of a common gram-negative facultative and aerobic, rod-shaped, non-spore formal bacteria belong to the family of the Enterobacteriaceae. The EA was actually named after Aerobacter aerogenes and was later included in the genus Enterobacter in the 1960s. It is also known to be resistant to antibiotics. It is a not so common and pathogenic bacteria that cause most of the opportunist infection. Disease. What disease does this organism cause? Well, EA is a non-common and pathogenic bacteria that cause opportunist infection. It generally does not cause disease in healthy individual. However, in cases where infection occurs, some symptoms may occur, such as eye, skin infection, meningitis, bacterium, pneumonia, and UTI infection. Host. Who or what is the reservoir or host for the organism? EA is generally found in the human GI tract, various ways, hygienic chemicals, and soil. The minimum optimal and maximum pH for EA is replication. It's 4.4 or 6.0 to 7.0 and 9.0. Transmission. How is this organism spread? EA is known to be a notorious hospital-acquired infection. Some of the infections caused by EA results from specific antibiotic treatments, venous catheter insertions, or surgical procedures. In addition, besides in the general population, healthcare workers, especially those who are intravenous drug abusers, such be considered subjects that could cause a transmission of the EA infection. Treatment. Avoidable treatment, management, cure. EA is actually highly resistant to antibiotics, so most drugs are ineffective at treating the infection. The best way to fight this bacteria is to have proactive prevention practice, active adherence to standard hand hygiene regimens, environmental decontamination procedures, controlled use of antibiotics, and aseptic insertions, catheters, and implanting devices can help reduce the transmission of this bacteria. This last page is actually my reference where I pull all my resources from. This is pretty much it. Thank you.